as a starting point while we're waiting for others to join. You could read through the discussion prompts again. I mean, what I just gave in the plenary was really a high-level summary of this, and I want to make sure that everyone has a chance to read through this. And as you know, these breakouts are split into two parts. Because of the change for the breakout in this case, for this specific visualization and search session, we propose that we do 45 minutes approximately, really, on the first part of the questions, and then spend 30 minutes on the second part about, developing some ideas and recommendations for how might we questions. Okay. So I would propose that we get started. I see a couple of people on video, which is great, and I want to begin with the overall question. Sort of, like when we're thinking about visualization in the context of salad lists and thinking about a human salad list. Whether this is the HubMap one or the HCA one or the HTN one or something that combines all of those. When we think about visualization in that context, I can see almost as many opportunities and needs for data visualization as there are data types that will be incorporated into that atlas, because we need to be able to look at individual data types. There's a need for visualization across many different scales. And we think about having an overview for a whole body, going down to individual cells and their context. We need to think about visualization that integrates with analytical approaches, that integrates with query tools. So the universe of problems, visualization-related problems to solve in the context of salad lists is, of course, enormous. But and on top of that, we have also already done quite a bit of work as a community as a whole on solving some of these problems. And I just mentioned one of them, which is sort of standard single cell visualization using embeddings and scatter plots. That's a problem that has been solved over and over again. Certainly also leading maybe us to a question like, why are these problems being solved over and over again? Why can't we solve that once and then move on? But I don't think I want to go in, or we don't want to go into that question specifically today, but something we should keep in mind as we're having this conversation today. So I want to invite you, if you have any comments, and like in this general discussion about visualization, there are prompts in the green box, and we have quite a few of them. But if you feel like there are other topics that we should add to that list, please, could you add them now, and maybe briefly speak up if you're planning to add something, so we know what these additional topics are. And then I would suggest we start with a conversation about, should we look at all data at once? Is that our ultimate goal? Or should we also maybe start by looking at individual data types or individual scales first, and then talk about how to bring it all together? So I want to open the floor to you. You're as much part of this as I am, and invite you to comment with discussion prompts or suggestions on where you want to start the conversation.
Nils, this is Anoop. We're not doing introductions, right? We're just going straight into discussion.
It is up to you. I have not managed to actually join one of these breakouts early enough to see what happens at the very beginning. As you might have noticed, I sound probably like a total newbie to this, but if you want to do introductions, I'm happy to do that. I think
some people have and some haven't. But anyway, so my name is Anoop Mahurkar, and I represent the Brain Initiative, a BICCN network, within which particularly we manage the Multi-Omics data coordination center for that. And so we have some visualization tools and some analysis tools. And what I was going to ask you a question is that the prompts sort of suggest that we'll have one single tool. Is that an aspiration or is that a mandate?
That is a great question. I don't want to answer that. I would like to hear what other people are thinking.
I'm just going to continue the introduction. My name is Andreas Bückle. I'm a PhD student at Indiana University. I focus on data visualization and virtual reality.
Thank you. Is anyone interested in adding either an introduction or if anyone wants to comment on this question? Which I think is a really important question to talk about. You know, what is the endpoint here?
Do you hear us?
Yep.
Yeah. My opinion, I think it's going to be important to share. Several people can share the same data. So several different eyes, pairs of eyes can look at the same data and have some comments on collaborative approach in visualization. So we can have a kind of reference data set or a benchmark or the reference data set. So we more can know what we talk or communicate about.
Yep.
So that's I feel it can be important.
That's a very good point. I think adding the question, I think there were two things, like using a reference data set as well as collaborative visualization or visual exploration and the ability to communicate. Thank you.
And also, so we can there can be several contributors on a kind of common data set we can refer to, kind of landscape of data sets we can talk about or have names on and we can refer to.
Okay.
So,
thank you.
Yeah.
I think, does anyone want to comment on the suggestion of collaborative visualization and the ability to contribute sort of to a reference data set?
So I have a question. This is, I'm Fiona Ginty. I'm from the HubMap group. I'm based at the GE Research Center. So we've been heavily involved with the multiplex immunofluorescence process for the last 10 years or so. And so I'm more focused on the protein side of things. But in our HubMap project, we're combining sequential sections of skin, healthy skin, so multiplexing 25, 30 sections, combining those into 3D and then comparing those. We also have a micro CT image of the tissue samples as well. So we have a 3D and then we also have the 2D that's reconstructed to a 3D. So we're sort of in the process in the early days of figuring out all the visualization of that. I'm a biologist, but our computer vision person on our team, Yusuf Al-Kafahi, he's not here today, but he's starting some early ideas on how to integrate that data and visualize, but it would be really nice to know what tools are out there already and that we're not reinventing the wheel either. So I suppose my ask is, what is the sort of state of the art, and are there tools already out there, even in the outside the biomedical space, that could be used?
I think this is an excellent question. Thank you. I think about creating essentially a map of the tools that are out there to work on the kind of data that we're planning and hoping to integrate into these atlases. I think this is also, by the way, a very actionable thing. Right? One could imagine writing a review paper or setting up a database, or there are databases out there. I really like this. I think something that we should note as that's certainly how might we actually get this overview and understand like what do we have and what do we need? Where are the real missing pieces in the puzzle? Because a lot of problems have been solved as an individual problem. Thank you for that. I like that.
Yeah, just to add to that, we are also working on imaging data and integrating imaging to get positional information while harvesting tissue samples. So I'm at the Medical University of Vienna Division of Anatomy, and we work on body donors to get CT and MR images, also maybe ultrasound integrated in the process of harvesting the tissue already.
Excellent. So is this actually a data type that, I'm not sure if we had ultrasound on our list of data types, but it's certainly something we should add. Thank you. Can I hear a couple more thoughts on this question of, you know, are we ultimately going to build a single thing? Like is there going to be the Google Maps for the human body for Cell Atlas? I personally have an opinion about this, but I would love to hear your thoughts on that. If that's something that your vision, sort of, that you have as your vision, because that is sort of the public view on this, like, oh, well, we'll have the Google Maps of the human body, essentially. But I'm not sure if this is really the reality that we'll be creating here.
This is Aaron Horning here from Stanford. I like the idea of there being a single map. I also am not sure how realistic it's going to be able to do that. And also just logistically being able to put it all together. It's a lot of different data types. There's issues with ontology, with the CCFs. And it's not something that we should shy away from, but it's something that I think that logistically, it's not going to be something that we're going to be able to just go ahead and just do once. It's probably going to be different groups are going to put different things together, and then we'll come to some sort of, oh, we like this aspect of this one, we like this aspect of that one, and then possibly put it together. But I just see logistically it being kind of a difficult task to do, especially with the different scales. There's a lot of difficulty there. And in the previous meeting, in the breakout sessions that we were having, some of us were talking about, smaller scale things, and then other people are talking about larger scale things. And so that discussion needs to happen first before we can build this integrated map. So there's a lot of logistics going into this. It's going to make it difficult.
Mm-hmm. Okay, so yeah, I fully agree. I think the issue of scale is a really critical one here, in understanding, like, coming to some sort of agreement. What are the scales at which we want to operate, or which we want to map the data, and how to integrate all these things is absolutely critical before we set out to build any sort of integrative, majorly integrative tool across, maybe that would be used across multiple atlases.
I would like to join the previous speaker and also pointing out that one tool would also have to serve 9 or 10, 12, 13 different kinds of target audiences. So of course, if you have just one tool, is a nice start, but this one tool would have to serve so many different stakeholders. I think it would be absolute overkill to put all of this functionality into one single tool. Although the thought certainly sounds nice. I further think that I think many tools that appear to be one tool are really many different ones. So if we think of Google Maps, of course, it feels stepless, the way I zoom in, the way I retrieve certain information. But really, those are 3 or 4 very different types of software put together into one interface so that you feel like you're using the same tool. So yeah, I think putting it all into one tool is unrealistic.
Thank you for the comment. I think one thing I do want to actually loop back to is this point of stakeholders, which is a really, when you think about data visualization, who your users are really determines what you're building, right? And because it's visualization, in a way, provides a very easy way to interact with an atlas, like, say, compared to using a statistical model, a lot more people actually will access this information through visualization tools and the like. So I think as part of our conversation here, we should think about the stakeholders for which we'll be building visualization tools. There will be researchers who are experts in building these atlases. There will be other biologists who want to just use the atlases and want to understand them. There will be, hopefully, some high school kids at some point who want to look at what we know about the human body at sort of the single cell or tissue level. And there are many other potential users. So I think having a good overview of, like, who we would be creating these tools for is helpful, and something we should not forget to do before we decide on what to build and with what priorities. However, I also want to point out that in that same vein, well, this is not a new problem, right? There are a lot of public biomedical resources that are accessed by a wide range of users. And I think we don't have to reinvent the wheel here and come up with our own list. So I think this is a pretty universal problem.
Is the Allen Brain Atlas a good example of something that's sort of multimodal and already out there? I don't know if folks are, I'm sure everyone is familiar with the Allen Brain Atlas, but also it depends on whether you want to have analytics as part of the atlas or the visualization functionality. So that, of course, would add some extra complexity. But maybe the Allen Brain Atlas is just something sort of already built. It's maybe a good starting point.
I was going to say, again, getting back to the end user, we really need to understand what the end users are going to use it for and how they're going to use it. But I think we need people with human-computer interface expertise. I mean, I happen to own a software company, and you usually have to tell the software program engineers to actually engage with these people and customers early, because otherwise you design something which people just don't end up using. So it's going to have to be a complex team of people with different expertise to develop software at that level.
Absolutely. I think, yeah, go ahead, sir.
So, I would imagine that the use case for our tool, independent of the stakeholders, is going to fall into two very broad categories. One would be communication. It basically just means I have a very concrete question in mind that I want answered. So I go to the website, I use the tool, then I have the answer within a couple of actions. The other broad category, I think, is exploration. So for example, Nils, you mentioned a hypothetical high school student who is interested in biology and, you know, wants to look up cell types in the kidney. And so this kind of user goes in, spends probably more time in there and needs a lot of interactive tools to change what they are seeing right in front of them. So I think these are two very broad use cases that are going to be relevant in the long run.
Yep. I mean, I think this distinction, since you mentioned exploration, I mean, the sort of just having a visualization on an Atlas website that summarizes information that's available, that's one thing, but really exploratory tools that allow users to discover patterns, unexpected patterns, that allows them to generate hypotheses that can really guide their own research. That's a completely different beast, obviously, from you know, building some basic tools or visualizations that are used to explain what's available or to explain how the data was generated. So I think that's very critical, like the stakeholders, to distinguish these different use cases for data visualization in the context of the cell atlas. Okay, so yeah, go ahead.
And one last thing, I mean, you know, for the multiple types of users that we're going to have, I mean, I see it being easy enough to just kind of set filters for like, oh, this is a high school person, oh, this is a graduate student. You know, we need different filters for different types of data that are presented. It can all be based on the same sets of data, but you know, just more simple visualizations or something like that.
Yep. I agree. I think ultimately the data obviously needs to be the same, but sort of how, like the route you take to access it, that can be very different, and whether that's implemented through filters or just different tools, I think is an important question that we need to address. Okay, I want to not forget about our prompts and our list of things here. So just to remind you, we have until 2:55 overall. So in between, we should take a 15-minute break. And I think what I would like to do is go through, or try to go through the first couple of prompts in the green box, and see where that leaves us. And then take a break and come back. So I think we'll probably, did I get that? Yeah, 2:55. So I think we want to maybe take a break around in maybe 25 minutes or something, so around 2:20, and then come back for the second half, just so we have that on our radar. And I need to write that down. I don't know. I also already want to thank our scribes. I see them typing away here. Thank you so much for doing this. This is incredibly helpful, and your work is absolutely critical to this breakout. So I want to go back into the discussion prompts and maybe talk a little bit about. I mean, maybe I can share my personal view on this question of you know, is there a single tool or are there going to be many tools? And I think there will be many tools. I think the integration of building a single integrated visualization tools to view and explore a tissue atlas or cell atlas, as the ones we've been discussing here, is something that maybe will happen in the future at some point in the distant future, once we have really fully understood all the problems that we need to support with visualization. But for visualization, in my experience, is often the case that when you build a great visualization tool that provides users with a lot of inside understanding of the problem, you're going to make that visualization tool obsolete because people will learn enough about the data that they're looking at in order to write tools and develop algorithms that can solve many of these problems computationally. So I think that is in a way what I would like to aspire to, building visualization tools that make themselves obsolete over time so that we can automate a lot of the tasks and answer a lot of the questions algorithmically rather than requiring a human being to look at that. And I say this as a visualization researcher, that's my bread and butter. But I think that is really ultimately the goal where we want to get, at least with these exploratory visualization tools. So let's talk about first steps and what integration could look like, right? So I'm sure we have a very diverse group here in terms of the data that you are familiar with, and there are probably also some people who are looking at all sorts of data because they look at this more from a visualization and methods development point of view. But the first prompt here, how would we want to achieve integration? Like what is it that we want to look at? Do we want to look at derived features? Do we want to look at the image data behind some of the derived features? And of course, in some cases, it's maybe not an image, it might be sequencing data that is sort of the raw data here, or primary data. So and so we were talking about integration across assays in that case, right? So rather than integration across, say, different tissues as a starting point. So where do you see the opportunity here for integrated visualization, and maybe also define what you mean by integrated visualization in that case?
Well, I can speak for at least what we're doing in skin. So with the micro CT image, there are some sort of visible features in the stratum of the skin, and so that can then in turn be aligned to the H&E or the autofluorescence image actually of each of the sequentially stained tissue samples. So from that perspective, going from that macro scale to micro scale, it's at macro feature level. There's no other easy way. Blood vessels do actually, the sort of tiny blood vessels that are in there, they're a good matchup point as well. And then from there, you can start to use the H&E to layer on the cellular data, but we haven't gone as far as RNA either. So, but at least our goal is to, right now we can do the micro CT at a sort of stratum blood vessel level to the H&E, the autofluorescence image, and then ultimately bring the cellular data into that as well.
Yeah, I think that I agree with Fiona here, and also it's something that's echoed in the things that the CCF group yesterday was talking about also, that in order to think about how to develop these CCFs and understand where all the different cell types are, you have to start from the macro scale and like have an image of a normal body or something like that. And then you go down to like CT images of like, and then you build a model of what you expect, like let's say the liver to look like. And then, within that model of what you have of a normal liver, maybe even a diseased liver, something like that, then you start putting on these layers of, okay, these are the different cells that we have data on. And then, this is the, you hover over a single cell there, and this is the overall gene expression profile that you have of it, and this is the cell type and things like that. So I think it's definitely going to have to be, we have an idea as to what we expect this to fit on, and then we layer on our data onto that at whatever scale we have.
Great, thank you, Aaron.
Well,
so, yeah, go ahead.
How are we going to define normal when it comes to, say, a liver or a heart or anything else? I mean, because there's a lot of variation between people, which is completely normal. It's just a sort of a thinking exercise, because whatever the normal for the images won't necessarily be related to, say, the 10x or whatever, when a small piece of tissue is being digested or the imaging has been done. So it's just something I think we just need to think about at some stage.
No, that's true. Absolutely.
And I think, Mark, your comment is really well taken. When Jason and Peter and I discussed what we want to discuss with you in this session, this question, you know, what is the normal? Can we have some sort of a reference for tissues? Like, what should it look like, right? And how do we use that for visualization? That's the part we want to discuss in the second, 30-minute plenary at the end, because we think that is relevant not only for visualization, that is relevant for search and many other things. So I really like this, and we should probably have a little bit of a conversation about this. Like for those of you who are maybe coming from the like genomics world, like we're so used to comparing everything to a reference, and it's so easy. Like all of our visualizations for genomic data are based on a reference usually, not all, but most of them. And it's very easy to visualize, differences, like say like variants in a genome, because a genome is, it's so like there is no representation, or you can choose the representation you want to use for whatever visualization problem you have. But like when you're working with an image, like you have spatial information that you need to maintain. There's already, like, your screen is full of data. How do you like, then show what is maybe different between two tissues, or even how do you synthesize a reference tissue representation and incorporate that in your visualizations? It might not be possible at this sort of anatomically correct representation that we're thinking about maybe, but it is certainly something that could be done once you have the data abstracted away from the, say, the tissue image. When you're talking about embeddings and things like that, that's where I think we have a lot of potential to bring in reference information, like how things typically look like, and then how does your whatever sample you're studying look like compared to that reference. I think that's a really, really interesting question, because in genomics, this is like, that reference is what drove, I think, most of the visualization work for years, right? The genome browser was the one way of thinking about, how the genome looks and how it works and how we visualize.
If I could add, I think there is a distinction between what you would call a reference versus a canonical reference. So a reference could be just something we agree upon, and there is no canonical human. In terms of language, thankfully, but in terms of language, one thing that's been adopted in some clinical consortiums is just to use the term healthy. So not normal, we just stay away from that term. It's healthy and the rest of us.
That's a great point. Thank you, Todd. So, however, I guess what is the definition? I mean, I don't want to go deeper, but then it sort of the question is what is the definition of healthy? But I guess in the end, what you pointed out, the reference versus something that we just agree on versus a canonical reference, that is important. And I think when we're talking about genome sequences, we have the same problem, right? There is like that reference does not exist, right? There is no one out there who has that reference genome. So it's something we agreed on to use, and there's a process to agree on what we want to use at any given point in time, and it changes over time as well. So I think you're making an important point that we need to.
Even for that reference genome, like, I think that HG38 was based off of 13 individuals or something like that. So I mean, then it was just all agreed upon. So it might be that we start with a reference that all of us agree upon, and then eventually it sort of changes when we find certain variations that are important to consider and include in the upcoming references as well. But I don't know that we need to worry too much about it, but it is, of course, a very important aspect of building this map.
Yep. Okay. Thank you for the comment about that. So, and since we're just talking about the references, I think a question about the CCF and how the role that visualization, the CCF will play in visualization is certainly warranted. So I think there are some people, Andreas, I don't know if you're involved in thinking about how to visualize the CCF for HubMap. There might be other people here who have thought about how a CCF or Common Coordinate Framework would be used in the context of data visualization. And in a way, it is one of the references we can use, for example, to navigate the space. But Andreas, I don't know if you have any comments on that pointer. What does it look like?
I can talk a little bit about how we are currently using the CCF for very early prototyping work. Actually, what you see behind me is a part of a tool that we created for HubMap called the Registration User Interface, where tissue mapping centers can indicate the extraction site of a tissue sliver, which you actually see right here, inside a kidney in this case. And this is then later registered against the CCF and displayed in our CCF exploration user interface. But of course, again, this is very early prototype work. And it kind of leads back to an earlier discussion we had about the importance of user testing and interface design. So we are trying to work together as closely as we can with different stakeholders at different TMCs to find out what exactly it is that our end users expect from a tool like this. And I expect this to be, of course, a multi-year process that's going to involve a lot of informal and formal user testing. Especially with something as complex as the CCF that is nowhere near finished. I hope that answered your question to a certain degree.
It does. And in a way, I already knew the answer because I've seen your work. Are there others who have thoughts on the use of common coordinate frameworks in the context of visualization?
Yeah, I have some ideas on it.
Yeah, go ahead.
Because, I pictured, okay, so my background is tissue engineering, and I've been working on building 3D CAD models on a cellular level of authentic tissue architecture. So my thought is, some of the stuff I've been working on could be applied to this common coordinate framework system. There's, the structure that I really think would be important, besides the vascular tree system, would be the basement membrane system. So if you can map the model the basement membrane system, it would give you a 3D encapsulation of all the cellular architecture of an organ. And then you can create coordinates along the system, and then use that as reference points where you'd be actually, well, I mean, if I could share my screen, I could show you what I'm talking about if y'all have time.
We could do this. I don't know if we can do that. Let's see. Who's the host for this session? It's not me. Can you want to try to share your screen?
Yeah, let me try and see what happens.
It looks like I would be able to do it. Yeah, looks like.
Okay, so here's like a kind of drawing that kind of shows, this is showing the lung, it's showing the basement membrane outline of here's an alveolar sac, and you can see you have your airspace and your blood vessels here. Here's an actual CAD model of the lung. The red represents the blood flow through the alveolar system. The blue is the airspace, and here you can see a TM of a similar area in a primate. You can see where the blood cells are. You can see here's the airspace, and here's not the scale, but this is like a printed version. You can see so the basement membrane would line these locations like this, surround the blood vessel and surround the airway. And when you have something like that, and you could go to the heart and create these kind of structures, either reconstruct it in 3D and get the encapsulation, or you can use special stain. And here you can see in this model here, you can see these are the chambers. It's just a section out of this heart here that's been stained with silver nitrate. Here is where the cardiomyocytes will lay. Here is where the blood will flow through the capillaries right alongside these cells. So if you have something like this, then you can, hold up. You see, then you can apply coordinates.
Yep.
You can apply coordinates right here to the surface, like shown here. You can apply the coordinates to the surface of those areas that I demonstrated here. So you can apply the coordinates, and you can have tables with these coordinates in them, and you can list information, or I'm not going to go into detail, but you can actually use this to reference in detail, you know, where the locations are in three-dimensional form.
Excellent. Thank you for sharing that. Are there comments on what William just shared?
I'm trying to get out of here now.
I actually have a question about how you construct those 3D models. So I saw that you had a bunch of hand drawings on one of your slides. So when you actually build those models, are those being hand modeled in a software like Maya, or are they being modeled computationally or parametric?
No, they're actually modeled from STL data, but depending, if it's, some of it is done through, you know, serial sections that have been reconstructed, and then image captured as STL and then converted to like a mesh, 3D mesh, and then converted into NURBS models for surface rendering. And, you know, that's how, you know, would see ideally having it done.
Thank you.
Great, thanks.
For me, this highlights two things. So first of all, the importance of recording positional data as metadata with all the tissue samples that we are taking. And the second is that geometrics, morphometrics might already have some of the tools that we need to compare image data and to position other data within a coordinate framework or within other image data.
Yes, that's a very good point. This is not an area that I'm particularly familiar with. Is there anyone who has experience with that, who could point us to approaches that other communities have?
Right. So what, you know, one of the things I didn't really go into is that once you create these layers, that you can, you know, you can add information to the actual model surface. And when you do that, you know, the method I've developed, you know, you come up with a code that can be embedded into that model, and that code translates out to like a table with any kind of data you want to have in it, you know, and it could be the cell type, the genes that are in that specific cell, or, you know, whatever data that might be, you know. So yeah, there's ways to do that. And then when you can go to, you can just type in the 3D coordinates that you're interested in. It'll take you to that location in the model, and then on top of that, it'll show you the embedded data for that location. So yeah, I think that would be an ideal way to do it, you know, because the way I've developed it was for 3D fabrication using like femtosecond laser technology, so that you would be able to go specifically to any part of the model and, you know, apply, you know, whatever type of, you know, application you want to do to the fabrication process for that.
Okay, great, great. Thank you very much, William. I think we are running a bit short on time here. If you want to take a break at 2:20, we should talk about some of the other topics that we have on our list here. And so we have already touched a little bit on the synthesized image problem, but where I think some really interesting questions lies, you know, about how do we build visualization tools, and how can we collaborate in building visualization tools for tissue and cell analysis. So I'm curious if people who are in this breakout, if those of you who are building visualization tools or are integrating visualization tools into you know, websites and data repositories, if you could maybe share a little bit about your experience with doing that, and sort of what approach you've taken. Are you building, trying to, you know, reinvent visualization for this kind of data? Are you reusing existing visualization frameworks and tools? So where was your starting point if you are doing such work? And since I don't hear anyone speak up, I can maybe briefly share what we have started to do for HubMap. As the approach that we have taken so far is to build a new visualization tool, or almost a suite of tools that we're designing specifically for two purposes. So one major driver for this tool or framework that we're building, which is called Vitesse, is to make it embeddable in all sorts of different contexts. So it needs to be possible to embed individual components in the data portal user interface. It should be possible to embed these components and reuse these components for the design and implementation of new exploratory tools that are really also potentially tied up and hooked up to some analytical tools. And sort of the ability to really reuse components in a common framework, and where the connections between different views are defined based on kind of entities and events on entities in these visualizations, such as a cell has been selected, a gene is selected, or changes were made to those things. So that's one of the drivers. The other driver for that approach that we've taken is we want to be able to integrate all the new data types that are being developed, and we would like to be able to integrate them at a level that goes beyond just saying, well, here's an image, here's a TIFF, or here is a, here's an embedding of some cells, or here is a, some peaks in some single cell ataxic data, because all of these assays have much richer information and sort of the one thing that you look at in the end. So the ability to integrate the rich information about these assays with the visualization is one of the drivers for us to say, okay, we don't just want to pull, like existing visualization components and then put them into our portal or into tools. We really want to try to come up with a framework that is flexible enough to tie together many different data types, because that's really where the exciting new questions lie with these tissue atlases and cell atlases, that we have so many different modalities that we want to bring together. And at least in our experience, and it's maybe just our opinion, that you can also only integrate data in a visual way if you have tools that actually support sort of seamless navigation between views and perspectives on the data. So that's what we've started to do for HubMap. It's also clear to us, of course, that this is not the only way, not the only set of tools that we'll be using going forward. We'll bring in other commonly used visualization tools as well. But then this leads us to the point, like, how would we integrate some of these more advanced tools that have a rich set of features with some lightweight components that we have maybe embedded in the website? That is something that we have not solved yet and might not be able to solve at all from a technical point of view. But here I'm curious. So we're definitely taking a very flexible, modular approach to building visualization tools. And definitely everything has to be web-based, because we don't believe in building desktop applications for anything. So do you have thoughts or comments on that when we're talking about architectures and how to build applications?
Yeah, hear me?
Yes. Yeah.
I've been working on visualization tools for pretty long time, and I'm missing a kind of a data set. We have to make our own test data, so that's a problem. We would like to have a large access to a large data set that other people are working on also, for example, have made in research on, so we know about the data, and so we should not look at the tools isolated, but it's a part of the data, and so to say. So we should not separate tools from the data. The tools are in some way derived from data.
Yep. Thank you. I think you made two comments. One is like having data sets available to use for like to test tools, I guess, to develop tools against. And the other is in a way, I mean, like that you have to think about like analysis and visualization as a single thing, and not just, let's analyze some data and ultimately someone will come and look at it and say, hey, let's build a visualization tool. I think if I understood you correctly, you're advocating for doing this, that's a process needs to go hand in hand. Do others have comments on that?
I want to follow up what you said earlier, Niels, about using a modular approach. I mean, that is, of course, the way to go. We are using DeckGL and Angular in our lab. Although the prototyping, as you can see again in the background, we did with a video game engine called Unity 3D, which allows us to prototype user-friendly systems very fast. So for me specifically in my PhD, it's very important to get to informal user testing very fast. And so any software that we can use that allows us to mock up a interactive user interface quickly is very helpful for us. But that, of course, sometimes might be at odds with the wish to make something that is more flexible and more expandable in the long run. And I think that is probably a contrast that many labs have to navigate when it comes to implementing visualizations.
Mm-hmm. Thank you, Andres.
I think that you know, the stuff that we've been doing in my group is seems much more primitive compared to what you guys have set up already. But you know, what we've done so far is we for the HTN project that we have, we've got you know, numerous polyps in the colon, and so when we take out the colon for a colectomy and then we look at all of these polyps, we want to know how far away they are from each other, and you know, what portions of the colon each polyp comes from. So what we do is we take a picture of everything, and then we stitch the pictures together as best as we can. And then what I'd like to develop is some sort of interactive interface for people to be able to just hover over where, you know, there's a polyp there. And so what information can we get from that polyp? And what does that tell you? And then also get out information about all of, you know, that entire 2D structure is what we're working with, and see how far away these polyps are from each other, and use that as a model for tumor growth. But being able to somehow integrate that data that we have into one of these frameworks, where it is in 3 dimensions, and we can overlay it onto what, you know, a reference, quote unquote, colon looks like, would be really helpful. And I think, you know, really informative also. It just adds another, you know, 3-dimensional layer to the spatially informed models that we can make for tumor growth and things like that.
Great, thank you, Aaron. So I think, well, we've talked about a number of different questions related to practical aspects of designing and building visualization tools. I am wondering. So I think there is one. Well, there's one more question on here that we could touch upon now, but I think maybe we want to leave this until after the break because we've been talking for a while. So what I would suggest is that we're going to take a 15-minute break now until 2:40 or whatever hour it is at your place. So until it's 40 past the hour. And I come back, and then we have, I think we only have 15 minutes to talk a little about what we could actually do and what we want to accomplish here. I want to get back to our agenda. We have great notes. Again, I want to thank the note takers. I think that's Dave Miller, who has been hacking away at that, and possibly others. So again, thank you so much for this important work. So the second part, I guess, is related to our, or where we should focus on these how might we do X questions, and really think about some concrete outcomes and some things we could actually do together, or at least some things that we could identify that people could collaborate on to move data visualization and for SALADless forward. So I did have a very, very quick look at the notes, and there are a couple of how might we questions in the notes. And I'm honestly completely open to which ones of these you want to discuss. I do not want to be the one who's talking all the time. I'm happy to facilitate the discussion, but certainly would like your input on that. So based on the, I mean, if you look at the notes from the previous session or the prompts, you know, do you see how might we do X questions that you would like to discuss a little bit and talk about? And if not, I will propose one to talk about.
I think the one that we kind of touched on was the one of the questions that we could possibly talk about was how might we generate the reference, or how might we add data to the reference, something like that. So I think that's one thing that we could work on.
Yes. So I like this one a lot, and that's actually since everyone liked that question so much, that's basically one thing I think we want to focus on actually in those 30 minutes after this breakout when we're coming back to our visualization search group. So I think I would reserve that for later because that is more or less the topic for that part.
Just on a note on that,
yeah.
Immunology has a habit of being particularly difficult in this area because it's highly dynamic.
So say it again, the last word. It's highly
dynamic, i.e. cells move around all the time. And where they are today is not where they are in a few minutes later.
Yes, a very good point. I think that's a comment that we should probably add in terms of challenges related to having a reference is in immunology with a lot of movement of the cells. Aaron, sorry for postponing that one, but that's a really important one.
Maybe here's one that we don't need to postpone. Maybe nobody cares in the big group, but I think how do we collaborate to build visualizations is really I think of interest to everyone. Or it should be, right? I mean, everyone is trying to do it, and we, like you said, don't want to reinvent the wheel multiple times, but maybe it has to be done differently for different consortia.
Exactly. So thank you for putting that out there. That would have been my proposal for something to talk about is, can we identify a visualization use case that we could collaborate on across consortia? Can we identify sort of a minimum viable product that would be a collaboration across consortia? And yeah, Anu, please go ahead.
Yeah, I'll let you finish. What I was going to suggest is we never address the prompt about the consensus seems to be that we're building an ecosystem where there'll be multiple tools. So then for an ecosystem to work, you have to have defined APIs so that tools can be written to the APIs to query the data. And that'll be something we haven't discussed a lot about. And so I don't know if that's important for the people on the call here, or maybe it's not, but I just want to propose that.
Like building a whole ecosystem with APIs, et cetera, to, okay. Yeah. I mean, it plays into this question that Philip and I posed. You know, how could we build something that is a collaborative effort? Because that would assume that every consortium, or at least those who want to participate in this effort, have the same or at least very similar APIs to get the data for these visualization tools out of their data repositories and databases. So but by the way, I figured out that we have until 3:05, so we have a little bit more time. So we have about 18 minutes. I don't hear anyone else with any proposals, so I think we should talk about that question. What is some low-hanging fruit in terms of collaborating on visualization tools across consortia? I wanted to ask you a question though, and I should have probably asked this question at the very beginning. Before we jump into that, can you maybe, you have these cool collaboration or participant tools where you can raise your hand or say yes and no. I was actually wondering who is actively involved in building visualization tools and designing visualization tools or integrating visualization tools into data repositories or for any of the cell lists that are participating. Use your button on you can click the green check mark when you click on participants. You can see that because I think that's sort of an important question actually. If there are a lot of people here in the room who are users, we can talk a lot about building tools. And if most people just care about having a tool, that's a very different story. So I see 1 no, thank you for that. And 4 yeses, okay. And a lot of people are still focused on the coffee. Good, so it's a small group of people who are building tools and designing tools in this room, but that's fine. So I think I want to break this down into two parts, this conversation. Let's start with the use cases. What is a cross consortium use case for visualization? What are some of the tasks that we anticipate, or that those of you who are not building visualization tools and maybe have a looking for tools would expect to be able to what are the visualizations that you would anticipate to see across all of these different salad lists as they are being built right now? So I think anyone can chime in on that.
I guess the lowest hanging fruit, but maybe still difficult, is integrating, and I think that has been discussed elsewhere too, so I don't want to be redundant, but integrating spatial imaging, whatever you want to image, let's say protein, with single cell sequencing data that are not spatially derived, right?
Mm-hmm.
And I know that at the H10, there's thoughts about this. You're aware of those, I think, a little bit.
A little bit, yeah.
Yeah. So, I mean, I think that obviously, I think it should go beyond that, but that may be something that every consortium needs and that could be kind of addressed together.
Yep.
And the other one is actually integrating multi-omic data, right? Because there's ATAC-seq, which is positional data with gene expression, which could be somewhere downstream. And how do you integrate those two data types and visualize them together? Is sort of an open question.
Right. And so maybe for both of you, the question that I have is, with this information, the data sets that you're thinking about right now, are they derived from the same sample? Are those assays that are sort of multimodal? I mean, I guess you can do this with imaging and then the omics on that, but because I think those are two very different questions. Because if they're not from the same data, then the question is like, is there a reference that you can use to map these things together somehow?
At least for the omic data, both are true. In some cases, it's coming from the same cell. And in other cases, coming from the same tissue, but from different cells in the same tissue. And so
yes.
I could see it similar to how we do things on oh gosh, what's that one where all the TCGA data is, where you basically get to select the data sets that you're interested in. You could select the the samples that you're interested in, and then be able to make comparisons based on that. I see that being an interesting use case too, to see, oh, this population of people has this sort of spatial structure, and this population has it like this.
Mm-hmm. Okay, so are you thinking about a GDC, or what is it, genome data commons, or there's so many of those these days.
Bioportal.
Bioportal. Okay, Bioportal. Yeah. Okay. All right, Phil, did you have when you mentioned that integration, spatial versus non-spatial, you thinking about stuff coming from the same?
I mean, I agree with you that it's very different, right? But I think the tougher challenge to me is coming from different sources. And maybe I'm on the outside, so you can correct me there. But I think if you have it from the same source, you have internal references, right? If you want to, and I think what we want to do eventually is, maybe compare data sets from HCA with HubMap, right? And so you would need this, this means to do, having to cross-reference different sources.
Exactly. Yeah. Okay. So let's. I think that's an excellent use case. That, I mean, we are facing this in HubMap. I guess H10 is facing that. I don't know what other consortia on this call, or in this group. So let's talk a little bit about this. So there is an assumption that we have a representation, a visual representation for the spatial information, which it could be an image, right? It could be a microscopy derived image. It could be some multiplexed microscopy or something like that. There could also be if it's an assay that's not really an image, but extracts information in a different way, if it's not an optical thing, like imaging mass spec data, which will also give us a spatial thing. Now, the things that the resolution for those is probably not at the cell level at the moment. So that's the image. And then we have the corresponding like non-spatial information that we can project. Or we can look at that in a, you could imagine having, if you have single cell ataxia, this could be a genome browser. So we have at least two different views on the data, the spatial view and then a non-spatial view most likely. So in order to build something like this, we obviously need the visualizations for both of these things. And they have to be able to interact with each other so that we, for example, can see, you know, if we use the spatial information to say select a region in an image, that we can highlight, for example, non-spatially derived information that should be in that region of the image and vice versa. So for something like that, this implies that there is some communication between the views and the user interface. There also implies that they can operate on the same data source somehow. So there has to be some common data model that describes the relationship between those two, at a minimum 2 data sources. There could of course be more than 2, right? I mean, but let's maybe focus on the simpler things first. So, and if you want to think about that, like how would we accomplish something like that? We need to have some agreement about APIs that we can query. Now, there could be some translation layer. Ultimately, we're dealing with the same entities, cells, transcripts, whatnot. But we need to be able to get that information. And that's at some level that assumes that whatever NIH-HCA and HubMap and all the others are building APIs that we can access from these visualizations. Then I think on the view level, when we want to do sort of the integration, the visualization tool, there is a need to have a mapping between the two data types that we're dealing with. And that is something, I mean, that's a universal problem, which I think each consortium might have their own solution to doing this. And I guess it depends also a little bit on the data type. But what I'm wondering about here is, you know, when we're talking about retrieving data from an API and visualizing it, we're talking about having some sort of an agreement on the entities and how this linkage would work. So there has to be a lookup somewhere to tell me, you know, this cell or these transcripts correspond to whatever region in the other view. So I think that's where in my experience as someone who has built a lot of visualization tools, that's where the real challenge lies, actually, in getting everyone on the same page in terms of these data structures, the entities, and how we can learn about these connections between the two data sets. So I think that's where a lot of the conversations need to happen first, and then the more visualized, like the more visualization specific problem, or more visualization specific problem is, well, like, you know, let's assume we're dealing with a large number of entities, or we're dealing with large images. Like, we need to have components that we can use to actually visualize this information. Although that is something that can be solved in a way almost in isolation, and people have solved this in you know, multiple times. Like, there are a lot of large image viewers. Can they be embedded on in any website? It's a different question, but many of them are somewhat flexible. The same thing with, you know, large scatter plots, for example, or genome browsers. We have these things. I think we really, if we want to collaborate on that stuff, we need to think about the data first, and being able to sort of have a common data model that we can think about and that we can operate on. So I'm curious if anyone has comments on that, like whether we can accomplish that, whether we can get a data model. And I unfortunately couldn't make it to all the other sessions that I meant to attend, but I think there were discussions about this at this meeting.
Yeah, if we share data, we should agree on what metadata we should follow the data, some kind of standardization, or how data are collected. So yeah, in principle, it's possible to reproduce the data somehow.
I'm having a hard time hearing you. Can you get closer to the microphone? It's a little muffled.
Okay. let's see. Do you hear it better now?
That's slightly better, I think.
Okay, okay. Now it's okay. No?
It's okay. Give it another try.
Okay. I will try to be a bit practical. If you share data, we should somehow agree on a minimum metadata. What data is following? How is data collected? I have received high experience from receiving some image data, and often I'm missing the necessary metadata.
Okay, so
that is maybe what is important when you really work on using the data to develop software. You need metadata.
Right. So I think what I'm hearing is you're advocating for, I think what most of us sort of agree with, this sort of we need good metadata about our experiments, and we need to be able to, we can hopefully use some of that to do these mappings. But I think where a lot of our, at least if I'm looking at what HubMap is doing at the moment, I'm not sure where HCAN and other consortia are in terms of defining the metadata. It's at a very different level. It's sort of, we want to describe our assays and our samples, but I don't know if our metadata is currently sufficient to sort of automatically infer what can be, for example, be displayed in the same visualization, and what can be linked and what cannot be linked. So we have only 5 minutes left, and then of course we have more time. We can bring this question back to the main group. In particular, I think because for the harder use case that we just talked about, this question of like, if you have data that is not from the same sample, like what is the reference framework that we're going to use to link it together? I think that fits perfectly well into these remaining 30 minutes that we have with everyone. Mm-hmm. But I did want to, yeah.
Mm-hmm. And I have a question. So maybe it just sort of feels for me a little abstract at the moment. Is there a good data set that we could already draw on that could be collaborative, and maybe that's a goal to just define what that data set is? And certainly we have data we can share, but just maybe that would sort of concretize some of the actions or the follow-ups.
Yeah. So I'm happy for others to answer that question. I mean, I can just say what we have started for our work. We have used published data sets that were not from HubMap, because HubMap is not at a point where we have, like, data from like even the same tissues across a lot of assays. I don't know what others have done. I mean, does HCAN have a multimodal data set ready that would be useful for this?
There may be. I know that you and Peter Sorger and others have been discussing this source for multiplex imaging data sharing, and there will be at least one single cell RNA sequencing data set that would go with that sample.
Yeah.
So that could be done in a way beyond just HubMap and HCAN, but I think that's exactly what's needed, right? I was about to ask the same question. Is there anything people could start working with?
Yeah.
To figure out how to connect the two?
Yeah. I can give a recommendation. I mean, we have, for our work, we have used a data set by Stan Lenarson's group. They have something on their OSM fish data, which is spatial information. They also have individual cells derived from that. There are expression profiles for those cells. So that is a very, very cool data set if you're trying to build something integrative, because you really have it all. But it's small, and it's not something that's been generated by any of these consortia. So I think we have to potentially wait a little bit as well. But I did want to acknowledge, and Mike had a question, and a very good one, actually. So maybe, Mike, you want to go ahead and raise that?
Yeah, I had to step away for a bit, so I don't know if this got covered or not. I've heard some talk about, could we go from whole body to focusing on individual cells? But sometimes the biologist wants to know about cells that are distributed through the body plan. Show me all epithelial cells regardless of the organ, or show me all neural crest derived cells. And I want to compare them even though they're not in one anatomical location. So is there talk about supporting that kind of use case?
That is an excellent question. We didn't talk about it, not while you were gone, and also not while you were here. So it's something, I think this is sort of a question about almost a definition, like what defines data set, right? Because I noticed in HubMap, we get data from all sorts of tissues, but then, do we define a data set as like, well, it came from that sample and maybe it happened to come from a kidney or from colon or from whatnot. But you really you're talking about indexing data in a way, like by cell type, for example, and being able to run a query and say, give me all, epithelial cells and show me visualization of those, of course, with the associated metadata where they came from. But I think this is a really, really, from a visualization point of view, it's not different from saying, well, I want to have all these cells that are actually co-located in the same tissue versus those that are spread out. But it's sort of a question about having the infrastructure in place in a data portal to enable these queries, to pull it out. And that, I think, is again, it gets us back to the, like, how do we get to the data problem that we, I think, really need to solve before we can tackle these more exciting visualization tasks.
Yeah. Nils, so the BRAIN initiative has generated some interesting multimodal data, including multi-omic as well as imaging data associated with it.
Right.
So I just propose that as one more data set to look at.
Okay, can you post that in the chat channel, or put it? Maybe someone can put it in the notes. I can put a link to the data set that we have been playing around with. I'll just put it in there. That would be helpful. I think collecting something like this is useful. So we are now it's 3:05. Is everyone okay with leaving this breakout and going back to the bigger breakout to talk about how we incorporate reference information, how we maybe build references and incorporate those into visualization and search? Or is there any last or any final comment from anyone?
I was just wondering, Nils, if you want to, now that we have this wet lab free months ahead of us, if there's a way to get something like that started using these data sets, just, I don't know, everybody throws that word jamboree around or whatnot, but if one wanted to give it a try, right? See how can we connect those data sets and who has the easiest, best, most intuitive way to do that.
Yeah, so you're thinking to do this across consortia?
That would be the hope, yeah, and then see,
I guess there is a final plenary across all breakouts. I think that would be a really interesting question to bring this up there. Like to see, can we identify a tissue, I guess, that has been assayed by a lot of different projects for that to make sense, or a cell type or something like that. I like that idea. Since all of our months are wet lab free, not too much has changed for us, but I think bringing that as a question to the larger group would be very helpful. I would be delighted if we could come up with a joint data set that we all can use to develop our tools and that we can sort of use as a test case. I think where things get a bit tricky is, are people going to be willing to actually load these data sets into their portals? HubMap by definition doesn't do cancer, but I think that's something we could work out, and it would be fantastic to have that. So I really like that point, and I think we should bring that also back to the next 30 minutes that we'll start now.
Is there a central repository intended or already in place for data sets across all sites, just so that it would make it easier to find those data sets rather than go to each atlas?
No, at the moment, the conversations we had with HubMap and HCA in the context of curation was sort of to try to annotate data sets in a way that they are compatible with the different data repositories, so that we could, in theory, load HCA data into HubMap and vice versa. I don't think there is any joint, there are no plans to build a repository of repositories or something like that. It would have to be it could be a Google Drive folder or Dropbox folder, but probably not, because a lot of data is identifiable. So it would have to be in a certain place, which, by the way, Austin, I guess leads us back to the question of like data use agreements, et cetera, et cetera. So certain data, cannot be readily shared. So it's probably slightly more complex than just putting the data in one place.